Hi, I'm Barbara Ray. I'm the Nature Education Coordinator for the City of Dublin. In this session, we will consider the most recent updates of the novel coronavirus in animals, resources where we can direct people who have questions, as well as look at the role animals play in the development of treatment and vaccines. Throughout human history, people have been afflicted by zoonoses. That is, diseases transmitted to people from animals. Rabies has been known since around 2000 BC, and the first written record of rabies is in the Mesopotamian Esmuna Code of Babylon in 2300 BC. The code dictated that the owner of a dog showing symptoms of rabies should take preventive measures against bites, and any Babylonians that had a dog transmit rabies to a person had to pay a fine. These diseases, zoonoses, can be acquired directly, as when a person is bitten by an infected animal. They can be acquired indirectly. For example, a person bitten by an arthropod vector, such as a mosquito or tick, that has picked up the pathogen from the host animal. People can come into contact with pathogen-bearing fluids produced by an infected animal or consume food contaminated with animal-derived pathogens. Some zoonotic agents maintain an ongoing reservoir cycle in animals or arthropods without becoming a permanent established disease in humans. Other agents are what we call species jumpers, and they derive from an ancient reservoir life cycle in animals, but subsequently establish a new life cycle in humans that no longer involves an animal reservoir. And currently that's where we are with COVID-19, and that in general it's not infecting animals, it's just jumping directly from human to human. Of course, there are some animal incidental hosts, and we'll talk about those too. So we know that primates have suffered illness and death from disease acquired from other animals since primates first showed up on the planet, which was about 60 million years ago, near the beginning of the Eocene epoch. This is about the same time that the first horses and opossums showed up, right about when T-Rex was becoming extinct. And of course, horses and primates have changed quite a bit over time, whereas possums have remained largely the same. But anyway, the first primate fossils have been found in North America, Europe, and Asia. And the rise of primates coincided with the evolution of flowering plants. So this is a timeline of primate and hominid evolution. The earliest proto-simians, pictured on the upper right of the slide, were animals such as the Plesiodaphiforms. And most of us have seen this human evolution chart, or maybe even visited the exhibits at the Smithsonian in Washington, D.C. But humans have contended with zoonotic disease from the very beginning. Human hunter-gatherer populations currently suffer, and presumably have suffered for millions of years, from infectious diseases similar or identical to diseases of other wild primate populations. However, the most important infectious diseases of modern food-producing human populations also include diseases that could have emerged really only within the past 11,000 years, following the rise of agriculture. We infer this because these diseases can only be sustained in large or dense populations of people, and those didn't exist anywhere in the world before agriculture. A good example is leprosy, which is caused by Mycobacteria leprae, which has recently undergone historic analysis, and raised interesting questions about its origin and how it spread. It was first recorded in humans around 600 BC in India. The molecular evidence points to it being far older, however, possibly originating in Africa during the Paleolithic period. Another bacteria that has gone through genetic analysis is Bordetella pertussis, which is the bacteria that causes whooping cough. That's originally thought to have passed to humans from a similar species of pertussis found in domestic animals, and the molecular evidence suggests that it's been around since before animals were domesticated. It may have evolved from the bacteria B. bronchoseptica, which was present around 2.5 million years ago, with a preference for infecting hominids. This kind of makes pertussis a neat little story of a bacteria adapting to fit changing hominids as they became human and evolving specifically to fit the human niche. Pretty much similarly to the way, say, lyssaviruses have followed bats through 400 million years of evolution. So they've really perfected their place in bat species, just like pertussis has in humans. The World Health Organization defines zoonoses as diseases that are naturally transmitted between animals and humans, sometimes through an intermediate non-vertebrate host. And that may be a virus, a bacteria, or a fungus. Some examples are West Nile, Lyme, lepto, rabies, and TB. But a key to understanding prevention of these zoonotic diseases is to know about both the ecology of the pathogens and the immunocompromised status of individuals. And this is what people are really learning a lot about now with coronavirus worldwide. This is a USGS graphic depicting the human overlap with wildlife and domestic animals. And of course, some diseases come specifically from both sets of animals, and some diseases can come from wildlife or livestock equally. And this graphic depicts how a virus might make the jump from just infecting people, say a rabid animal biting a person, to a limited outbreak where, say, a human actually was able to infect another human with the virus, but in a limited way with an animal vector, or a long outbreak where many animals and humans cycle, and then what is considered stage 5 becomes a human element. So human to human transfer, which the current novel coronavirus has managed to accomplish. So 60% of our diseases that are infective to humans are zoonotic diseases, and 75% of new pathogens are zoonotic. So basically the CDC publishes 6 out of every 10 infectious diseases in people are zoonotic, which makes it super important that we strengthen our capabilities to prevent and respond to diseases, and they recommend using the One Health approach. I've included this because One Health is the interface between wildlife healthcare, veterinarians, and human healthcare. It recognizes that the health of people is connected to the health of animals and the shared environment. So as human populations grow and expand, we're entering new geographic areas, living close to wildlife, or maybe domestic animals too, which allows diseases to have an opportunity to pass between the two. Certainly, we've disrupted the environment in tremendous ways, and disease can take advantage of that encroachment. And then we move people and animals and animal products across the globe. also giving viruses a chance to move around. So where did this thing come from? Actually, animals, of course, play a role in the SARS-CoV-2 virus, which is a beta coronavirus, or in other words, a bat coronavirus. It's one of thousands of viruses that bats carry. In a study with over 12,000 bats that were studied from Latin America, Asia, and Africa, 9% of them carried one or more of 91 different coronaviruses. And it's estimated that over 3,200 coronaviruses infect bats, and there's 1,400 bat species worldwide. So in a way, that 3,200 is really not that big when you're considering the sheer number of bats. But coronaviruses have lived with bats for a very, very long time, so they're really good at living in bats. One of the primary research that's gone on over the years with coronaviruses has been in horseshoe bats. Since a lot of ever since the SARS virus, researchers have been interested in these cave-dwelling bats because of their association with the SARS virus. Rhinolophus affinis is a horseshoe bat that lives in southern China. And back in 2004, a study was going on, again, to study this SARS virus. And back then, they were looking at what's called a bat sarbecovirus, RATG13. And that was from a 2013 study. But basically, these viruses had, the current SARS virus has a 96% genome identity that's the same as the viruses carried by these particular bats. And most researchers involved with these studies are very confident that the lineage from which SARS-CoV-2 has emerged has been present in these bats for several decades. So this is not new, it's just that the virus has made that jump to humans. So not many bats were actually found in the Wunan market when scientists were first trying to find the origin. Of course, it's important to find the origin of a new outbreak because that can often, early detection may help stop the virus in its tracks. And those genomics were not possible during, say, the avian H5N1 outbreak in 97 and 2003, and the SARS outbreak between 2002 and 3. But by 2009, rapid genomic analysis had become routine. And so the influenza pandemic and outbreaks of MERS, and H7N9 avian influenza in 2013, Ebola in 2014, and Zika in 2015 resulted in rapid sequencing and genomic characterization. And as a result of that, most of those diseases were halted pretty much in their tracks. But it's hard to infer reliable evolutionary history for coronavirus because the recombination rate is so high. So when they were looking at the bats, they also were looking at the people, and they took blood samples from people living near the caves who volunteered to share that. And 3% of the population they studied had antibodies to several of the coronaviruses with the same strains carried by the bats, which means somehow they were coming into contact with these bats, getting exposure to viruses, and then their bodies were launching immune responses. Some of these folks were sheltered in the caves, especially on hot days in these tropical zones in the summer. So they very likely had a lot of exposure to bats or bat guano, possibly without even really knowing it, and would have been able to build up natural immunity from low-dose exposure over time. So since these single-strand RNA viruses are highly recombinant, it wouldn't have taken a lot of effort for the virus to make a change once it reached a person to where there was person-to-person spread. And if the people who were carrying this virus in these local communities probably weren't necessarily making each other sick if they were all in clusters and already inoculated by the bats themselves. So these people travel, and as soon as we move a new disease around, we're going to infect other people. And in fact, part of the success of colonial Europeans coming to America was not their weapons, it was their diseases that they incidentally transmitted to Native Americans that wiped out thousands and thousands of Native Americans. And of course, the virus may have incubated in another animal, and because we typically have viruses use intermediate hosts, that was originally what everyone was kind of looking for. And the animal that came to mind because of similar ACE2 specific residues that are shared on receptors with both humans and pangolins, and because of the pangolin market, pangolins are the most highly trafficked vertebrate mammal, it made sense that potentially they were an intermediate host. But the residues are in the pangolin Guangdong 2019 sequence, and there's reasons related to this sequence of residues that it's like the ACE2 residues in humans. And they were also present in a common ancestor, which was that I mentioned that RATG13 that also came from the bats. But these residues were actually lost through the recombination in the lineage leading to the RATG13. So basically, those sequences have changed in pangolins, which it doesn't really make sense anymore that the pangolin would have been an intermediate host. And in fact, it makes more sense that this disease made a zoonotic jump right from the bats, because the key ACE2 binding residues are present in viruses circulating in bats at this time. So basically, at this time, there's no evidence that pangolin infection is a requirement for bat viruses to cross into humans, but it was a viable theory, certainly. So how do these viruses make the jump from an animal to another animal or a human? So one of the things they do is they drift. So small mutations occur, and this is how we identify the variant. So when they first identified, say, people in Washington State, how the virus all of a sudden traveled to North Dakota with the same variant because someone got on an airplane and went there. So it's possible to track which viruses are related, even from bats to people, because of these small mutations and who's related to who. But because RNA viruses are so highly recombinant or changeable, they also can become virtually unreadable in that all of their ancestry can change very quickly. So drifts are not that bad. They really just represent different variants or strains usually. A little more dangerous is a shift, and that's where several of the virus strands or variants change so drastically they form a new or novel subtype. So the new SARS-CoV-2 coronavirus is a new subtype that humans have never seen before, and that's why it's possible to become very sick from it. So here's a graphic showing both side by side. SARS-CoV-2 is a single positive strand RNA or ribonucleic acid, and it encodes for 10 genes. And the virus can enter cells by binding with ACE2 or angiotensin converting enzyme 2 through its receptor. The RNA viruses usually have high mutation rates compared to DNA viruses because the RNA polymerases lack what we call the proofreading ability that the DNA has. So it's difficult to make an effective vaccine to prevent disease caused by RNA viruses because they're so diverse and they keep changing. SARS-CoV-2 virus codes for 10 genes, and for comparison, the rabies virus, which is also a single-strand RNA virus, codes for only 5 genes. A lot of DNA viruses, like pathogens like herpes virus, smallpox, adenovirus, and papillomavirus, they code for hundreds or even thousands of genes. And this is a graphic depicting these spikes or S proteins that can bind to the ACE2 cells and enter a person's cells. So when we start looking at animals and COVID, obviously the virus has to be able to enter animals' cells as well. And it turns out that it does, because several species of animals have similar enough ACE2 binding points on their cells or receptors. So initially, back in the spring, a few dogs and cats in Europe started testing positive, and there were a handful of case studies. Then in the United States, our first COVID positive animals were actually a few tigers and a lion in the Bronx Zoo, which then spread to a few more cats. So interest was piqued in cats in general and big cats and what was going on. So these cats, it turns out, had a keeper that was an asymptomatic positive COVID carrier. So the person was spreading a lot of virus particle, and the cats were particularly susceptible to it. In the meantime, IDEXX labs started testing literally thousands of dogs, cats, and horses, and a few other species, but only a handful of positive dogs and cats have been confirmed through their tests, the CDC, and the USDA diagnostic lab. And generally what IDEXX does is when they got a positive case, they also ran 2 other tests. They might have run ELISA tests, but they also confirmed with PCR. And then the CDC would retest those samples, and if it was positive, then they would consider it a positive case. So we now know a lot more about which animals are affected and how they're affected, and of course continue to study this. So how are these animals shaking out? Well, basically, we now know that ferrets, cats, and hamsters are susceptible, and they can be easily experimentally infected, and that they spread the infection to conspecifics in laboratory settings. And of course, as it turns out, they're spreading it to conspecifics in both home and even some outdoor settings. Some of the studies are investigating non-human primates as models for human infection, because in order to come up with treatments and vaccine, we need some animal models who can also be susceptible or show infection. Some of these studies have looked at pigs, chickens, ducks, and mice, and they are very resistant and they don't spread the infection. So they're not good animal models for that. Some dogs can get infected, but they don't seem to be giving the virus to other dogs compared to cats and ferrets, which easily spread the virus to some of these other animals. So let's look at some of these species individually. In those IDEXX tests, and I think I found IDEXX had listed really less than 20 positive dog and cat cases that were also tested by the CDC. And the few dogs that did test positive had very low viral loads in the test result readings. They all had acquired in a home or a setting where they were exposed to either one or more positive family members. They rarely show signs. Occasionally a dog would show a sign, but most of these dogs didn't show any signs. They were highly resistant to the virus and developed antibodies fairly readily. And it doesn't seem that they spread it to other dogs or people in the home. Cats, on the flip side, are very susceptible. Some don't show signs, but some do. And they tested more often with a high viral load. They're definitely showing themselves to be infective to other cats, both in and out of the laboratory. So researchers definitely are doing continued investigation on cats, as they could turn out to be a silent intermediate host. And the biggest concern with any animal that readily can acquire the disease that we have is they also might become a reservoir and could give it back. Ferrets, mink, badgers are all highly susceptible. They develop signs similar to people, and unfortunately for mink especially, they get very sick very quickly, and once they start showing signs, they usually pass away within a day or two. And their illness looks very much like our most serious COVID cases in humans. Similarly, elderly mink and ferrets are more susceptible than younger ones. But they're a good model for vaccine development because they actually get the illness very similarly to people. So there's trouble in River City for mink. COVID-19 affects mink in similar ways to people, causing respiratory problems, again, worse in older animals. They show open mouth breathing, discharge from their eyes and nose, but again, they don't live very long. They usually pass away pretty quickly. On necropsy, their lungs are wet, heavy, and very red and inflamed. And a lot of mink have been examined here in this country since the Utah outbreaks by veterinary pathologist Tom Baldwin at Utah State University. And he says that what he's seeing is consistent with the signs of pneumonia and is matching up almost exactly with investigations carried out in Europe in the European mink outbreaks. So thousands of mink have died at fur farms in Utah and Wisconsin, but the first infections at big farms showed up in the Netherlands, and there's also been cases in Spain and Denmark. And over 1 million mink have been culled in order to try to control the spread. There is one documented case of mink transferring COVID to 2 workers on the farm, and this was determined by whole genome sequencing, where the only way these people could have gotten this variant of coronavirus would have been from the mink themselves. In other cases, workers brought the virus into the farm, and there are some cases in the Netherlands where cats on one farm transmitted the virus to nearby farms. So these were cats that traveled regularly between 2 or 3 different farms. And again, the mink showed the same genome sequencing from farm to farm, and there was no workers that worked at all of these farms, so it was just these cats. So in the United States, around the 1st of August, an outbreak occurred in Utah that's now spread to 9 different farms, and close to 10,000 mink have been lost to COVID-19. At this time, about 2,000 mink have been lost at a single Wisconsin farm. So no pelts are leaving these states, and the carcasses are being buried or incinerated. Each farm is under quarantine with just essential workers on site, and they're being directed to use the proper protective equipment at all times, of course. And these outbreaks are ongoing at this time. So how can animals help us end the pandemic? Well, in order to use an animal model, you need one that shows signs similar to humans. So that's where cats and ferrets come in. The University of North Carolina at Chapel Hill is conducting a study. They're awaiting results from studies with mice, ferrets, and monkeys that are being exposed to the coronavirus after receiving Inovio's vaccine. Since no species perfectly mimics human infection, testing a trio broadens the look at safety. There's actually some good news on the safety front, as the first animal data from various research teams is showing that there are no signs of the side effect called disease enhancement, which Dr. Anthony Fauci calls reassuring. In other words, enhancement is just what the name implies. Very rarely, a vaccine doesn't stimulate the immune system in the right way, and it produces antibodies that can't fully block infection and that also may make the disease worse. This happened in 1960, for example, with the failure of a vaccine for syncytial virus called RSV that is dangerous for children. And it's complicated efforts currently at vaccines that are trying to be developed for the mosquito spread dengue fever. Some attempted vaccines for SARS virus, which is our COVID-19 cousin, seem to cause enhancement in animal testing. Now we have recently reported studies in monkeys tested with different COVID-19 vaccines, including those made by Oxford University and China's Sinovac. But none of these monkeys showed evidence of immune-enhanced disease. So again, this is all good news. And also, having these 3 different models helps with the types of vaccines and how they target the spike proteins that coat the coronavirus. So we definitely have a need for some kind of a global system. We continue to be bombarded by novel animal pathogens, but there's really no ongoing systematic global effort to monitor the pathogens emerging from animals to humans. Such an effort can help us describe the diversity of microbial agents to which humans are exposed. If we can characterize animal pathogens that might threaten us in the future, we might be able to detect and control local human emergencies before they have a chance to spread into epidemics or pandemics. Wildlife surveillance can play a critical role in disease monitoring and prevention. So again, wildlife veterinarians, rehabilitators, zoos, anyone working with wildlife on a regular basis has the opportunity to notice perhaps some illnesses in animals that could become zoonotics. Certainly monitoring disease itself might best focus on people with high levels of exposure to animals. Hunters, wild game processors, veterinarians, rehabbers, wildlife trade workers, and zoo workers. So these folks become infected with animal viruses sometimes, and their infections can be monitored over time. Often they build immunity without even knowing it, and of course they can be traced with other people in contact with them. Animal caretakers have a lot of resources available now to guide through COVID-19 cases where animals and people interface. The CDC provides standard guidelines that are being followed globally. They update those as more information comes out and more understanding about how these viruses affect animals. The AVMA, the American Veterinary Medical Association, also provides a lot of resources online. And certainly we know now that we can and do spread COVID-19, particularly to some of our pet species living in the home who may have high exposure. So when folks have a positive COVID case or a positive exposure, so they might be asymptomatic, certainly we should separate ourselves from the animals we work with and have non-infected people working with those animals whenever possible to prevent the spread to our animals. So we need to be smart, we need to be safe to protect ourselves and our animals. Thank you very much.